We closed with the first introduction to the autonomic nervous system when we talked about that as a kind of three-box model with a preganglionic neuron whose cell body was located in the central nervous system, a postganglionic neuron whose input region was located in the peripheral nervous system in ganglia, and then finally that synapse of the postganglionic neuron onto the target. We'll repeat that today, but what we're going to do today is we're going to drill down and differentiate the two branches that we talked about yesterday, that is the parasympathetic and parasympathetic branches. The parasympathetic and sympathetic branches. Now, whenever you see parasympathetic, particularly vis-a-vis the heart, recognize that the role here is going to be to decrease the heart rate. And we'll talk about how this happens. We're basically going to be functioning through the so-called pacemaker cells and affecting the pacemaker potentials. The long and the short of this, where we're going to end up with this idea of the parasympathetic branch decreasing the heart rate, is we're going to work through G-alpha I, back to the G protein coupled receptors. And what we're going to do is we're going to inhibit adenylyl cyclase. If we downrate adenylyl cyclase, we're then going to reduce the amount of cyclic AMP. And if we reduce the amount of cyclic AMP, we've effectively reduced the amount of cyclic A that's available to bind to the HCN channels. So this is the parasympathetic branch. As we go through all this anatomy and all this rigmarole, in the end, on the heart, what the parasympathetic branch is going to do is it's going to reduce the amounts of cyclic AMP and work through the HCN channel. Yesterday, we emphasized that by and large, when we think about the parasympathetic and sympathetic branches, we think about opposite effects. We think about parasympathetic being rest and digest, and therefore slowing down the heart rate. And we think about the sympathetic branches being the fight or flight, or speeding things up. And in this particular case, the sympathetic branch is going to increase the heart rate. It's going to increase the heart rate by, again, working on the pacemaker cells. Again, the target is going to be the pacemaker cells. These cells will be activated effectively by binding to the neurotransmitter norepinephrine. Norepinephrine is going to again work through a G protein coupled receptor, and in this case, it will work through G-alpha S. We'll upregulate, we'll increase the amount and activity of the adenylyl cyclase to increase the amount of cyclic AMP. So we'll see that these two are working one against the other, but the final common structure that they're working toward is to affect adenylyl cyclase and thereby affect the amount of cyclic AMP and thereby affect the HCN channel. And we'll work our way through that. Now, in contrast to the parasympathetic branch, which is dealing almost entirely with the pacemaker cells, in the sympathetic branch, there's both the innervation of the pacemaker cells along with the contractile cells. And in the sympathetic branch, we'll see again working through G-alpha S, through something known as a beta-1 receptor. The norepinephrine is going to increase contractility, and this is more through a classic arrangement of phosphorylating channels, and we'll talk about that. So we'll draw this out sort of in a three-box model and just show it in generic terms. And then we're going to drill down into the absolute anatomy of these things and distinguish the anatomy of the sympathetic and the parasympathetic branches. And we'll walk our way through the anatomy. And what we're going to find is that the parasympathetic branch, for our purposes, and the heart, is going to have the preganglionic parasympathetic neuron cell bodies in the brainstem, the motor nucleus of 10. And then we're going to see that the sympathetic branch is organized differently. The sympathetic branch, again, will have its preganglionic sympathetic neuronal cell bodies located in the central nervous system, but now they'll be in the spinal cord, between T1 and L3 in the spinal cord. In both cases, whether we're talking about the preganglionics in the parasympathetic branch or in the sympathetic branch, those preganglionic neurons whose cell bodies are in the central nervous system send a process to the outside. They send a process into the periphery. In the peripheral nervous system, then, in both cases, the preganglionics synapse onto postganglionics. They'll then work through nicotinic acetylcholine receptors, always, pre to post. And that will then activate the postganglionic neurons, which in turn synapse onto target. And then on target, we'll release different kinds of neurotransmitters. We're going to release acetylcholine from the parasympathetic branch and norepinephrine from the sympathetic branch. And then downstream from that, we're always going to be working through G protein coupled receptors, and we'll walk our way through that story. So then, we're going to talk a little bit about some nomenclature here, and there's something called a muscarinic receptor. And this often gets a little bit confused. When we talk about a muscarinic receptor, what we really mean is it's a type of G protein coupled receptor. It's a type of G protein coupled receptor that binds to acetylcholine. This particular muscarinic receptor, when it binds to acetylcholine, the one we're going to talk about, will now reduce the activity of adenylyl cyclase through working through one of those trimeric GTP binding proteins called G-alpha-I. We've talked about these before. We talked about the endocrine system. We're going to see that then the sympathetic branch, at least in the heart, will now work against this. And it'll work against this by, in the sympathetic branch, the postganglionic sympathetic neuron, whose cell body resides in the sympathetic chain ganglia. That neuron now will innervate the target, in this case, the pacemaker cell of the heart. That neuron and the sympathetic branch will release the neurotransmitter norepinephrine. Norepinephrine binds to G protein coupled receptor. It activates the G protein coupled receptor, but in this case, it turns on our old friend G-alpha-S. G-alpha-S upregulates adenylyl cyclase. We increase the concentration of cyclic A. We now bind those cyclic A molecules to the HCN channel and increase its probability of opening, steepening the rate of rise of that generator potential produced by the HCN channel opening. And we'll walk our way through all of this. But let's start here with where we were yesterday. And the 3-box model of the autonomic nervous system. So I'm going to start up here at the top and just make the generic boxes that we had yesterday. And then I'll try to drill down and put this into context of the sympathetic and parasympathetic branches. So in our generic system, this is going to be the central nervous system. This is going to be a ganglion in the peripheral nervous system. And finally, this is going to be the target. And as we mentioned yesterday, the target could be cardiomyocytes. We're going to emphasize today the pacemaker cardiomyocytes. It could be smooth muscle, or it could be a gland. We'll get into the smooth muscle a little bit later when we talk about the respiratory system. We'll get into glands a little bit later when we talk about salivary glands and a little bit on sweat glands. But today's emphasis will be on the cardiomyocytes. The central nervous system, of course, contains neuronal cell bodies. The same story that we talked about when we talked about the nervous system. There are input regions here in the central nervous system. These neurons send a process out into the periphery that extends into the peripheral nervous system, where this so-called preganglionic neuron synapses onto a cell in the ganglion. And again, of course, this neuron has a name. And this neuron that I'll put in blue is said to be a postganglionic neuron. So this Y here simply meant to represent the output region of the preganglionic neuron. The circle is meant to represent the input region or cell body of the postganglionic neuron. And this postganglionic neuron then sends a process out to the target and synapses onto the target. Well, we can further generalize this, and we can say that whenever we have a synapse between a preganglionic neuron and a postganglionic neuron, regardless of whether we're talking about the sympathetic or parasympathetic branch, always at this site, we release the neurotransmitter acetylcholine. And always at this site, there are nicotinic acetylcholine receptors. Now, if you get down into the weeds on this, you'll recognize that these nicotinic receptors are not the same ones we see at the neuromuscular synapse. They're a little bit different. But they still respond to nicotine, and they still respond to acetylcholine in such a way that these are always ligand-gated, ionotropic channels. And then this blue neuron is going to now synapse onto the target. And this is where we begin to see differences. So let's go ahead and just go to the other panel. So that's the generic. Now let's, in purple here, describe the parasympathetic. Neurons and target innervation. And we'll repeat the same pattern. Up here is our box. This is the CNS. Now, if you take an anatomy course, you'll find that there are nuclei in the central nervous system, always in the brainstem, of cranial nerves 3, 7, 9, and 10. We're only going to deal with 10. But if you take a neuroanatomy course, you'll find there's a whole lot of parasympathetic and sympathetic innervation in the head, a whole lot of autonomic innervation that goes to the tear glands and the sweat glands, and particularly to the muscles of the eye, the ciliary eye muscles, which are used to focus, and also to the pupil, which determines the diameter, to the iris, which determines the diameter of the pupil. We're not going to get down into that, but recognize if you take a course in physiology, another course in physiology that emphasizes the autonomic nervous system, and certainly if you take any clinical courses, you'll dwell on that a whole lot. But what I want to do here is talk about the parasympathetic branch. So for our purposes, we're going to be talking about the brainstem. And specifically, we're going to be talking about something known as the motor nucleus of cranial nerve 10. Cranial nerve 10 has another name. It's called the vagus. And in this case, the vagus is really a good descriptor because the vagus comes out of the Latin. It means vagrant. It's basically the wanderer. And so the wanderer, these neuronal cell bodies here, whose cell bodies are restricted to the brainstem, these neurons send processes out into the periphery. And as before, they will go to a parasympathetic ganglion. One of the rules of the parasympathetic branch of the autonomic nervous system is that the parasympathetic ganglion is located near the target organ. Today we're going to see the parasympathetic ganglion for the heart. The parasympathetic ganglion for the heart is in fact the cardiac ganglion located in the wall of the atrium. But for our purposes right now, I just want to drive home the idea that this is a parasympathetic ganglion. It's out in the periphery. It has a neuronal cell body that receives a synapse from the preganglionic neuron. As above, the acetylcholine is the neurotransmitter, and the receptors are nicotinic acetylcholine receptors. Then this neuron sends a process out and innervates the target. Now, for our discussion today, the target is going to be the pacemaker cells of the conductile system of the heart. Again, these are cardiomyocytes. When we drill down and look at the neurotransmitter released here, we find that, as before, the neurotransmitter is acetylcholine. And this is a good place to bring up an old adage that we determined when we talked about the endocrine system. And we said, it's not so much the molecule that's released that determines the response, as it is the receptor to which the molecule binds. And so what we see here is this acetylcholine is in fact going to be inhibitory. Remember back here when we talked about, on the upper panel, we were talking about the generic system. We said these are nicotinic acetylcholine receptors. They're ligand-gated inotropic channels. They are permeable to sodium. So they're excitatory. So these excitatory synapses are located, always, 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 between the preganglionic neuron and the postganglionic neuron. The neurotransmitter is acetylcholine, but look over here. What we have here is, again, neurotransmitter being acetylcholine, but now it's inhibitory. And how can that be? It's inhibitory because it binds to a G protein coupled receptor. Whose effect is to downregulate. You can read reduce activity of adenylyl cyclase. Now, you remember from the endocrine system that the adenylyl cyclase is a really important enzyme. And what the adenylyl cyclase does is it converts ATP into cyclic AMP. It forms that phosphodiester linkage between the number 3 and number 5 carbons on the 5-carbon sugar of ribose. And this then becomes an important second messenger. I think you can see from what we've talked about so far what the story here is going to be. Remember, the HCN channel has to bind to cyclic AMP in order to open. So if in this case we reduce the amount of cyclic AMP and then we'll thereby reduce the number of open CN channels, what's that going to do to the rate of opening and the rate of influx of sodium into those pacemaker cells? It's going to reduce it. So that changes the slope of that ramp potential, reducing the slope of the ramp potential. Well, we can tell a similar story. If that's the parasympathetic branch, let's label this as the sympathetic branch. And again, use that generalizable 3-cell model. This is the central nervous system. The neuronal cell body resides in the central nervous system, but now in the central nervous system, this is going to be the spinal cord. In fact, it's going to be a particular region of the spinal cord located between the first thoracic segment and the third lumbar segment. Well, don't remember that, but just remember that these neuronal cell bodies are in the gray matter of the spinal cord, and I'll show a drawing on this a little later. Then this neuron, again, sends a process out of the periphery, because remember, we have to pertain to the upper generic structure. So the preganglionic neuron goes out into the periphery and synapses onto postganglionic neurons. Now, this is a sympathetic ganglion. There are some exceptions, but for the most part, these sympathetic ganglia sometimes known as a sympathetic chain ganglia, are located adjacent to the vertebral column. They're pretty much located right beside, a little bit beyond, but right beside the dorsal root ganglia. So they're out in the periphery. As before, there's a second order neuron. Then this now sends a great long process out normally that goes all the way to the target. Again, the pattern is the same. Preganglionic to postganglionic, the neurotransmitter is acetylcholine. The receptor is a nicotinic acetylcholine receptor. This nicotinic acetylcholine receptor is a ligand-gated channel. In fact, these channels are, as far as I know, interchangeable between the parasympathetic and sympathetic ganglia. Where things get to be different now is here in this postganglionic neuron. Typically, this postganglionic neuron synthesizes and releases norepinephrine. Norepinephrine is one of these molecules that's derived from tyrosine. Again, we mentioned it in the endocrine system. The norepinephrine is one of the major neurotransmitters here in the central nervous system and also here in the peripheral nervous system as part of the sympathetic branch of the autonomic nervous system. So the neurotransmitter here is norepi. And it would stand to reason that here on this target, that there must be norepinephrine receptors. And sure enough, there is a type of norepinephrine receptor called a beta-1 receptor. The beta-1 receptor is a type of G protein-coupled receptor, and it functions through G-alpha S to increase cytoplasmic concentrations of cyclic AMP. So one of the things I want you to recognize from these three-box models is that even though they're drawn in such a way that we have a sympathetic effect and a parasympathetic effect. And normally when we teach them, we teach about the sympathetic effect, we teach about the parasympathetic effect. One of the real hallmarks of the autonomic nervous system is they're never completely off or never completely on. There's always this yin and yang, this balance. Whether you're more biased toward the parasympathetic branch and therefore in rest and digest mode, or whether you're more in fight or flight mode and more in the thrall of the release of norepinephrine onto the targets. So that's the overall three-box model. So what I'm going to do in the upper panel is try to drill down, hold on to those two, and describe the anatomy of the system. And again, these will be pretty crude drawings, but what I want you to see is that from the boxes that we have above, there is in fact some real neuroanatomy. And try to convey to you from those boxes where the different boxes are in a biological structure. So let's first, on the left-hand side here, talk about the parasympathetic. System. So we'll again use our ice cream cone diagram, where this is the brainstem. And in the brainstem, there's this particular region, I'll show a sort of a black oval, called the motor nucleus of cranial nerve 10. And again, from above, the other name for cranial nerve 10 is the vagus, the wanderer. And one of the reasons that it has this name is that axons of neurons in the motor nucleus of 10 innervate post-ganglionic neurons in the thoracic and abdominal cavities. The one we're going to emphasize is going to the heart, but recognize that if you were thinking about the overall parasympathetic innervation of the body, you would have lungs and heart and gut and stomach and kidneys and all of the organs and liver of the thoracic and abdominal cavities innervated by these preganglionic neurons. And in all cases, the preganglionic to postganglionic synapse will be in a ganglion, and that ganglion will be located in the wall of the organ. The story we're going to tell here is the parasympathetic branch that innervates the heart. The neuronal cell bodies are located here in the brainstem. The neuronal cell bodies extend processes out in the periphery. They go out into the periphery in the structure known as the vagus nerve, also known as cranial nerve 10. They're the same thing. These neurons go out into the periphery, and in our story today, find their way here to a structure I'm going to circle in purple, known as the cardiac ganglion. All we've done here is replace the box on the upper series with a more circumspect region, which is the motor nucleus of 10. Those neuronal cell bodies, rather than being in a box, now are in a motor nucleus of 10. The nondescript extension of that neuron going out of the periphery now has a target. It has a postganglionic neuron target here in the cardiac ganglion. So, keeping with the theme, there's now going to be this neuronal cell body of the postganglionic parasympathetic neuron. So acetylcholine will be released at the site. Acetylcholine will bind to the nicotinic acetylcholine receptors and generate an excitatory postsynaptic potential. And if there's sufficient depolarization here, then we'll fire action potentials in this blue postganglionic neuron, which is now going to innervate the SA, let me do it this way, innervate pacemaker cells of the SA, sinoatrial, and atrioventricular nodes. So from the pattern above, you'll recognize that the this is the target cell. And so these will contain the so-called muscarinic receptors. The muscarinic receptors bind acetylcholine. But in this case, these muscarinic receptors are a type of G protein coupled receptors. So that's basically the anatomy. Again, always this three-cell chain, preganglionic to postganglionic to target. We drew that in the boxes. We have the same pattern here. Preganglionic cell bodies in the brainstem, synapse onto the postganglionics in the wall of the heart, in the cardiac ganglion, postganglionic synapse onto the pacemaker cells. Now, if we take the same pattern and we extend it to the sympathetic branch, we'll see that all along the spinal column between T1 and L3, that there are the preganglionic sympathetic neurons. So what I'm trying to do here is draw a column of cell bodies. These are the cell bodies of the preganglionic. The cell bodies, the preganglionic sympathetic neurons. So again, the next box down. Neuronal cell bodies in the central nervous system. I'm going to draw a cross-section of that over here. So where this is the spinal cord, this is our classic arrangement of the gray matter. What we find between T1 and L3 is there's a kind of outpocketing here of gray matter. This has a particular name. It's called the intermedio lateral gray column. That's a mouthful, but it's descriptive. It tells you that it's part of the gray matter, so you know it's going to be cell bodies. It tells you it's lateral, so it's off to the side. It tells you it's intermedio, so it's neither dorsal or ventral, it's in between. And so there are these neuronal cell bodies located here in a column of these neuronal cell bodies. And what I'm showing here are the cell bodies in cross-section that we see in longitudinal section, arranged, stacked one on top of the other. But if we take this pattern and rotate it 90 degrees and look at this cross-section, these cell bodies are located in the intermediolateral gray. These neurons have processes that come down across the ventral horn and go out in the ventral root. They take a bit of a circuitous route, but the most important part of this is that shortly after entering the ventral root, they leave the ventral root and enter this structure here that I'll show in blue. Known as the sympathetic chain ganglion. So if we come back here again, in the periphery, because remember the ganglia are always in the periphery, all along the spinal cord between T1 and L3 will be these segmental ganglion structures. They're bilateral. And these are intended to represent the sympathetic chain ganglion. Again, if you take an anatomy course, you'll find that these first two have neuronal cell bodies that are dedicated to innervating structures in the head. We're not going to get into that. What I just simply want to show you is the consistency here of the second three-box model and what we see here. So it's the same pattern. Again, the preganglionic sympathetic neurons have their cell bodies in the central nervous system, but they send their processes out into the sympathetic chain ganglia. Then in the sympathetic chain ganglia, those preganglionic, remember this neuron with the cell body in the intermediate lateral gray, again, is going to be a preganglionic. This is a preganglionic sympathetic neuron. The preganglionic sympathetic neuron, of course, makes a synapse in the periphery. In this case, it's going to form its synapse in the sympathetic chain ganglion. It's going to synapse now onto the neuron here, or if you will, up here, which is the postganglionic sympathetic neuron. But again, following the pattern of that three-box model, the neuronal cell body that resides here in the sympathetic chain ganglion sends its process out and now directly innervates the target. It directly innervates the target, and now the target is the pacemaker cell. So again, it's 1, 2, 3. Preganglionic to postganglionic to target. There's a synapse between the pre and postganglionic, which again will use the neurotransmitter acetylcholine, bind to nicotinic receptors in the same fashion we saw here in the cardiac ganglion. Same story. We generate action potentials in the postganglionic neuron. Now they have to, in the sympathetic branch, propagate this great long distance from right beside the spinal cord, in the neighborhood of the spinal cord, all the way out to the heart. Again, at this site now, the neurotransmitter the sympathetic postganglionic neurotransmitter is norepinephrine. Sometimes you'll see this epinephrine referred to in a generic term called a catecholamine. Now, we're going to see as we move through this and other systems that we can release the same neurotransmitter, norepinephrine in this case, and depending on what the receptor is, we'll get different responses. We emphasized that earlier here for the parasympathetic branch, where acetylcholine was the neurotransmitter both at the pre to postganglionic synapse as well as onto the target, but the difference in response depended upon the difference in the receptor. Again, when you look at the target, remember the target, the receptor in the target's always a G protein coupled receptor. So that tells us then from the upper panel that there must be a receptor here in the pacemaker cell that's a G protein coupled receptor that responds to norepinephrine. There's a GPCR, a G protein coupled receptor, that binds to norepinephrine. I'll abbreviate that norepi. So this G protein coupled receptor now is found in the target cell. So any questions up to here about the anatomy of these two and where the different neurotransmitters are and what receptors they bind to? Yes?
Is the pre-sympathetic neuron like part of the CNS, or it's part of the CNS and the peripheral nervous system?
So the question here is, when we talk about the preganglionic neuron, are we saying that's a central nervous system neuron, or are we saying it's a peripheral nervous system neuron? In fact, because it's cell bodies in the central nervous system, I would call that a CNS neuron. but just as I would call a lower motor neuron a CNS neuron. But the importance here is that the neuronal cell body where it's receiving input is in the central nervous system, in the spinal cord, in this case for the sympathetics, in the brainstem for the parasympathetics. But that process now extends out into the periphery. If we were to stand on the output region of the parasympathetic neuron here, we would walk all the way back into the brainstem. If we were to stand on the output region of the sympathetic neuron, we would walk all the way back into the central nervous system. But in this case, we'd walk back into the spinal cord. And again, if we were to stand on the output region of the postganglionic, we'll walk back to its cell body located in a ganglion. Either a ganglion in the wall of the organ, in this case the cardiac ganglion, or a ganglion aligned along the vertebral column, the sympathetic chain ganglion. Yes?
So where does the vagus nerve actually run inside the body? Does it just like run right down parallel to like next to your throat? Or
So where does the vagus nerve go? The vagus nerve runs along, I think it's the carotid artery. It follows the blood vessels. Again, that's an anatomy question. I'm not very good with them. And it will then follow that from the brainstem all the way down into the various organs, and branch and branch and branch. If you take a gross anatomy course, you'll find out. Anything else? Okay, if not, let's now leave this. And now, I'm going to erase this in a moment, but what we're going to do is we're now going to emphasize the concept of the target. We're going to see what's going on in the target. And much of what we're going to say about what's going on in the target is going to be review, because it's going to be the same story that we told in the endocrine system about G-protein coupled receptors. And we're just going to drill down a little bit farther here and describe a particular kind of G-protein coupled receptor. And then we're going to drill down and talk in particular about how these G-protein coupled receptors have their effect on adenylyl cyclase and how that directly affects pacemaker activity and directly affects the rate and rhythm of the heart. So we're going to do this again, imagining that we have a target as a pacemaker cell, and then imagine the parasympathetic and then sympathetic innervation of that pacemaker cell. Now recognize that as we draw this, that each of these pacemaker cells will receive input from both a postganglionic sympathetic neuron and a postganglionic parasympathetic neuron. But we'll separate these out into two boxes so we can follow a little better. So let's label this as parasympathetic response in a pacemaker cell. You can read, of course, cardiomyocyte. This box is meant to be, let me make it a little smaller. This box is intended to represent one of those pacemaker cells. Someplace up here will be the output regions of the postganglionic, parasympathetic neuron. If we look at these output regions, they're like output regions we've seen so many times before. There are these synaptic vesicles that contain, in this case, acetylcholine. There's going to be across the membrane here, a transmembrane protein that crosses the membrane 7 times. This is a classic G protein coupled receptor. The G protein coupled receptor in this case has another name. It's called a muscarinic receptor. The muscarinic receptor is a type of G protein coupled receptor. The muscarinic receptor is a type of G protein coupled receptor that binds acetylcholine. It has this peculiar name of a muscarinic receptor because it was named long before we understood it was G protein coupled receptor. It was named long ago, probably in the early part of the 20th century, because it would respond to alkaloids in a kind of mushroom, a kind of toadstool, most commonly Amanita muscarina, or muscaria. Now, Amanita muscaria is a mushroom we find around here. If you go into the mountains after it's been rainy for a while, you'll see these beautiful mushrooms. They have these red caps and little tan flecks on them. That's Amanita muscarina. And Amanita muscarina contains trace amounts of this molecule muscarine. And the muscarine now can bind to these muscarinic receptors. And one of the early days they found about this, if they were looking at the heart and they took this purified alkaloid, they could change the heart rate. They would slow down the heart. Because what they were doing is they were mimicking the effects of acetylcholine on the G protein coupled receptor, which we now call the muscarinic receptor. Well, that muscarinic receptor will now bind the neurotransmitter acetylcholine. And you pretty much know the rest of the story. You know that when the muscarinic receptor binds acetylcholine, it changes the three-dimensional shape of the cytoplasmic region of that transmembrane protein in such a way that this alpha subunit of the so-called trimeric GTP binding protein you know that this trimeric GTP binding protein is typically an alpha, a beta, and a gamma. And that typically this has attached to it GDP. But when this trimeric structure sidles up against the GTP binding protein with acetylcholine bound, there's a replacement of GDP with GTP. This now becomes an active alpha subunit. In this particular case, this is said to be G alpha I, and its function is to inhibit adenylyl cyclase, to reduce cytoplasmic concentrations of cyclic AMP. Remember, cyclic AMP is the main player in binding to the HCN channel, so this now is going to reduce the amount of cyclic AMP available, so it will reduce the probability of this channel opening, so we'll lower the rate of rise of the ramp potential. Let's have enough time to do any more. Oh, nope, okay, let's bag it here, and I'll come back tomorrow and do the sympathetic system.